Swayam Prabha. Digital India. Educated India.
Welcome. So we have discussed the anode break excitation phenomena, and we had one more thing to discuss regarding the Hodgkin-Huxley equations and explaining based on the phase plane analysis, that is relative refractory period. So if we consider spiking of a neuron, then immediately following the spike, we have a period of time that is called the refractory period. The beginning part is absolute refractory and the next part is called relative refractory. So, if we consider this as the voltage axis and this as the time axis, and let us say this is our V rest. At this point we have the voltage here. Now let us say we have some current injection at this time point, and let us say that it goes up, it crosses threshold, there is an action potential created that goes back down, and then gradually it goes back up to our V rest again. So the period in this intervening period where H has gone on to 0, to become 0, that is, the sodium channels are inactivated. So remember, when the voltage goes up high here, H time constant also reduces from the tau HV curve, and also the value of H at the high voltages goes towards 0. So when this voltage is coming down, by the time it reaches the bottom of it, H is already at 0, which means that none of the sodium channels can be activated, or even if the M's are open at that voltage, H will not allow sodium currents to open because H is 0. So that particular period where we have sodium channel inactivation, that period is called the absolute refractory period. So which means that in this particular period, no matter how much current we inject, we cannot get an action potential. So even if we inject a current here, because the sodium channel cannot open, none of the sodium channels can open almost, we cannot get an action potential. But in the following period, we have what we call is relative refractory, and that is a period where we have we need a higher current than usual to produce an action potential. So if in this state where there was no action potential, in green there was nothing preceding, there was basically silence in this green period before the action potential, then the size of the current required, this is the size of the current pulse, the size of this current required is much smaller or comparatively smaller than the size of the current that would be required in the relative refractory period to produce an action potential. So this height here, this height I, and let us say this is I0 before the spike. This I needs to be much greater than I0 to produce an action potential. So, and this gradually goes reduces to I0 when the neuron comes to V rest and beyond. I mean, basically when everything, all the gating variables come to their steady state value at V rest, we do not have any more relative refractory. But intervening period, we require a larger current to produce spiking. So, this also comes into play in some of the coding aspects that we will talk in the next part of the course. And the absolute refractory is very easy to understand in the sense that H is practically 0. So, there cannot be any spiking, and there cannot, I mean, just simply simulating the Hodgkin-Huxley, we can see that H is extremely low, and that region is what will be absolute refractory. But why do we have the relative refractory period is our question. So, in order to explain that, again we need to go into Hodgkin-Huxley equations and analyze it in the relative refractory period with our phase plane analysis. And since this has to do with a threshold or a production of an action potential, now with a larger current or a larger change in the membrane potential required to go beyond threshold, we will obviously now again consider the Vm reduced system. So, if we will again consider the Vm reduced system. So, if we draw the phase plane here, that is, we have V on this axis and M on this axis. We have our m dt equals 0, which is the M null-Klein, which is, as in our previous lecture we saw, is going to be the same, and this is our V rest here. And at V rest, when there is no external current, I externalize 0, and M, N and H have their values at the steady state values at V rest. Then if we draw the dV dt equals 0 null-Klein. So, this is the dm dt equals 0. Now, on top of it, the usual dm dt equals 0 null-Klein. Then we have this behavior as we have shown earlier, where this is this particular node, the lowest one, which we will call R, the resting node, which is stable. Similarly, the next node, which we will, let's say, mark in red, is the saddle node. This is simply a recollection of what we have done earlier. And then again we have another node, which is a stable node here at the m equals 1 value and a high voltage value around plus 53 millivolts. So, in this kind of scenario, we have a threshold that is determined, as you know now, by the stable manifold coming in from m equals 0 axis, and we have, if we consider this line along the resting membrane potential, wherever it cuts the stable manifold, the yellow line cutting the red manifold, say stable manifold, that's where the threshold is. So, voltages, initial voltages through current injection reaching anywhere on this yellow line before the stable manifold will go back to resting membrane potential, and anything beyond it will go along this line along this the unstable manifold and go up to that stable node onto the top right, and will produce an action potential. Now, if we consider the middle of the action potential or towards the end of the action potential, we have H and N different from what it is at rest. So, we know that H was at let us say we have H infinity VR. I should use a white. We have H infinity VR, as we saw earlier, is around 0.6 or 0.59. Now, what happens is, if we consider that H is more or less fixed at the values it reaches towards the middle of the action potential, that is when H has dropped down to about 0.1, let us say. I mean, that is what we tried to explain, that when we have the H values, when we have the voltage reaching a very high value, our H is reducing, going to 0, and then it is trying to get back up, and that is, let us say it is starting from 0 and then going back up, and let us say our H has reached around 0.1 and is staying there. Then we can plot the corresponding dV dt nullcline or the V nullcline, that is this particular dV dt equals 0 curve or the V nullcline with the new value of H. That we will see is that there is a shift upwards in the dV dt nullcline. So, what happens here is that this has reached here and follows that sort of same pattern. So, essentially we have the resting membrane potential or this stable node almost at the same place, whereas the saddle node shifts and is still a saddle at a higher voltage, and as the saddle node gets shifted to a higher voltage, it also drags along with it the stable manifold to a higher value or to higher voltage values. So, our saddle node is moving from this particular point initially to the new value, because our dV dt nullcline has shifted upwards, and the corresponding stable manifold with the saddle node that provided the threshold, that also moves to the right. And so, as you can now see, if we track the yellow line, the threshold of the system has increased by a few tens of millivolts. In fact, what if you simulate this, you will see that it goes to about around minus 40 millivolts instead of the minus 56 millivolts that was there earlier. So, that means now we need a larger current to move the neuron from V rest, effectively V rest or close by, to go beyond the threshold and produce an action potential. So, here remember we are moving, we should be drawing this on a yellow line. Remember, when we inject a current at V rest, we are moving along this yellow line, and that is effectively starting the system at a different V value, and that V value required is much higher to cross the threshold now. So, this is what we can say is effectively happening during the relative refractory period. So, in this period, even though the VR is at or the voltage is at rest or very close to rest, the same values of VR before the action potential do not reflect the same condition of the system. Remember, it is only VR. So, the previous history of the system, which is the lowering of H or sodium channels being inactivated and H staying there, makes the system behave in this manner. And this phenomena allows us to see the relative allows us to explain the relative refractory period. So, with this particular discussion, we will come to towards the close of our first section, and we will discuss a few points that we have made so far. Is that the most important points is that action potentials are all or none events, and that's because the neurons have a threshold, or rather a true threshold. We have something called absolute refractory, we have something called a relative refractory, we have something that is called the anode break excitation, which we will see later as a post inhibitory rebound phenomena. That is, a hyperpolarizing current is essentially an inhibitory input, and this post inhibitory rebound, that is basically an excitation or spiking post an inhibitory period, is because of the ABE phenomenon. And this also plays a role in coding of, let us say, stimulus offs, or stimulus turning off can be signaled with this post inhibitory rebound, and there are many other applications or concepts that can be explained with the post inhibitory rebound phenomenon. And of course, we have understood in this section the Hodgkin-Huxley set of equations and the Hodgkin-Huxley reduced set to explain the above phenomena. So, going forward, we will be requiring all these things in our understanding of coding by neurons. For example, if we talk of the absolute refractory and relative refractory, what this says is that there is a natural bound on the maximum possible firing rate of neurons, that they will naturally saturate because of a period following an action potential that is a period where no action potential can happen, that obviously bounds the number of potential that can occur within a limited period of time. So, essentially the rate of action potential occurring has a max bound. So, and we will see that rate of action potential is going to be a measure that will be often used, and that has been often used by researchers in neurosciences to understand coding performed by neurons. So, the other two, that is all or none events and true threshold behavior, will essentially allow us to model the spikes as in the spike trains as a sum of events, or allow it to be modeled as a point process. So we will be discussing these points later on. So the size of the action potential would not matter, but they will be simply dots or events in time, and so that tells us that we can have action potential strains to be modeled as a point process. And this, of course, we have already mentioned the post inhibitory rebound in terms of stimulus off periods and so on. So since the rate of action potentials is an important measure of response, it also becomes important to model the firing rate with simpler models of neurons. So in the Hodgkin-Huxley equations, we require to model the ion channels, their opening and closing, and the behavior of the different kinds of gates, activation and inactivation gates, which involves a lot of computation, and it requires more involved modeling, which also requires us to estimate the kind of ion channels that are present and their properties on every type of neuron. So if the generation of action potential process is not crucial in terms of the questions that we are understanding, and if the exact timing of the action potentials are not as important for the questions that we are trying to answer with our modeling, we can reduce the Hodgkin-Huxley equations to much simpler point models of neurons. So for example, if you think about the action potential process, the M, H and N, if we remove that from the Hodgkin-Huxley equations, we had already discussed that, which is the leaky integrate and fire model. So since the firing rate or rate of action potentials are important, then all we need is the number of action potentials that are occurring and not the entire process of action potentials. So all we need is using this idea of a true threshold that neurons have. We only require to simulate the voltage only up to the threshold point, and beyond that we don't need to consider modeling the voltage waveform, because that requires modeling the ion channels. So if we remove the ion channels in the leaky integrate and fire, we only need to define the threshold and know when an action potential occurs. Because they are all or none events, that is sufficient. However, the exact time is not going to be correct in terms of the real neuron, in the sense that because the upstroke till the threshold even is involving the M currents or the M activation gates of sodium channels, we will have the voltage reaching threshold slightly later based on simply the capacitor, the charging of the capacitor, and the active M conductance is removed. So, but if that timing is not important, just that there would be an action potential is sufficient. And in these cases, what we model in more detail is the I external going into the neuron model. So if we can get a better idea of what the input currents are onto the neuron, then our probability of firing or firing rate over time can be estimated to a very good degree, and we can understand variety of things, as we will see later on in the lectures. So, and these can be extended further beyond with more ion channels, little more ion channels explaining some adaptation phenomena, which is more of the relative refractory period like phenomena, that makes action potentials go further and further apart as if we have repetitive firing. So, these issues are covered in your textbook with using the spike rate adaptation and adaptive exponential. There will be a small discussion on this in your reading materials, and leaky integrated fire of course is important, and you will have exercises in modeling them also. So, from here the next stage is that, as we had discussed earlier, that we will now be recording activity from neurons, that is trains of action potentials, and there will be events occurring, either stimuli or some behavior or some other phenomena where would be occurring parallelly, and we would from those action potentials try to understand how those action potentials encode that phenomena or that stimulus and so on. So, that will be our second part of the course. Thank you.